Quick announcement before we get started. There should be a really fun seminar this evening on gene drives, whether we should or shouldn't do it, over in Kramer Hall, I think 238. Jim Collins, if I remember correctly, from ASU is going to be talking about sort of the idea of using CRISPR and CRISPR-Cas in animals that you could potentially be releasing in the wild. So that should be a lot of fun. He will also be giving the biology department seminar tomorrow at noon on the loss of amphibians. As many of you may have heard, there's been a huge loss in amphibian, particularly diversity, and lots of species have been dying recently. So we'll be talking about that. So those should be hopefully interesting, at the very least, things to talk about. I'll definitely be trying to go to this one this evening again from 7 to 8:30. I think it's Kramer 238, but I'll post on D2L what that is. Today, we're going to talk about nasty viruses, or at least things that people think are nasty, after a quick follow-up on our Paramyxo and Rhabdoviruses, mostly because Paramyxo and Rhabdoviruses molecularly are extremely similar to the Filoviruses, which are what we mostly be talking about today. They are all mononegavirales, so single molecule, negative strand genome. Their structures are pretty pleomorphic. We've got our classic Paramyxo virus up here, kind of a blob, but still has a helical nucleocapsid structure on the inside of a membrane and membrane glycoproteins. The Rhabdoviruses have a little bit more defined structure, but still they're enveloped and have this helical genome on the inside. Most of these viruses are going to bind and fuse to the plasma membrane, and so they come in directly from the outside of the cell. There's no sort of receptor-mediated encytosis, which is necessary. The replication process and transcription process, these are negative strand viruses, so they have to be bringing a RNA-dependent RNA polymerase with them inside the cell. So there's always that enzyme inside the virion. So very different than any of the positive strand viruses that we've talked about, which now they can come into the cytoplasm and get translated. These have to be making their own messenger RNAs. And so that is, again, a big difference between these negative strand RNA viruses and the positive strand RNA viruses. What these all do is they make large amounts of their structural proteins by having those at the 3' end of the genome, and then this start-stop stutter mechanism. So start making messenger RNA, get to a certain sequence, stutter for a while, add poly-A tails, sometimes fall off, sometimes stay on and make the next messenger RNA, and in that process you end up with a lot more of the structural proteins than the non-structural proteins down at the end of your genome. How they get out, they're budding again at the plasma membrane, and part of that has to do with getting fusion that can happen between different cells. And then talked a little bit about measles and why vaccines are absolutely critical and why all of us should be vaccinated. And I haven't heard back from my PCP about me getting my booster, but I'm working on that as well. So, any questions on Paramyxos and Rhabdos? Sort of, again, I'm going to cover a lot of the same stuff and sort of comparative-wise with these filoviruses, because the filoviruses are basically just big Paramyxo or Rhabdoviruses. Other than that, they're extremely similar to each other. And again, this idea of the mononegavirales having single negative strand. The reason that people care about these filoviruses is, of course, hemorrhagic fever. And people get very scared about these kinds of things. So, and we'll talk about, at the end, assuming we get there, why this is and why in some cases we should be scared and why we should be a lot more scared about what I'll be lecturing about on Friday, which is flu that nobody seems to be concerned about. So key concepts again, these are big, long rhabdoviruses. They also have RNA editing in a very similar way to what we talked about with the Paramyxos and Rhabdos, stuttering that happens inside of genes that will give you different C-termini of your proteins. One big difference that you see in at least the Ebola viruses, but not in Marburg virus, is there seems to be some kind of transcriptional regulation, and I put transcriptional regulation here in quotes because it's about mRNA production. And usually when I talk about transcription, it's going to be DNA-dependent RNA polymerases. But here they're RNA-dependent RNA polymerases. So they talk about transcription in the text, but I kind of prefer to think about this in terms of mRNA production. And we'll talk about it a little bit more again when we get to this difference here. And then, of course, the other big difference is this hemorrhagic fever process. So yeah, we'll talk a little bit about the structure. This is probably one of the most famous electron micrographs of a virion. Pretty much everyone knows what this one is. I'll show you the one next time that almost nobody seems to know what it is, even though they should. This image was actually taken by F.A. Murphy in a building that I will show you about halfway through the talk today. I've actually had the great fortune to be able to meet with Fred in terms of him taking some of these pictures. It's not straightforward to be taking pictures of these really nasty BSL-4 pathogens, I should say, biosafety level 4 pathogens, in an electron microscope, because most of the electron microscope facilities are not in BSL-4 facilities. In fact, I think I know of one in the world where this is the case, and we'll take a look at that a little bit later on. Yes, and then I would love to be able to take pictures of virions like this, but we're not allowed to do that for very good reasons here at PSU. So inside the genome, large negative strands, RNA virus, again, not terribly different from the Paramyxos and Rhabdos. Talk a little bit about the different proteins that are involved, particularly those that are different between the Paramyxos and Rhabdos, and most of these have to do with that regulatory process, and also potentially for some of the disease-causing processes. RNA editing is extremely similar. Regulation is this thing that's different, and then we'll spend a while talking about disease because it could become a pretty big deal. And it reminds me that there is, in fact, a rather nice autobiography by Peter Piul. It's a Belgian epidemiologist, virologist, who started out actually working on Ebola and then pretty quickly transitioned to working on HIV. It's a really fun read, very interesting book. So if anyone has a chance to take a look at that. And some of the stuff I'll talk about here comes from there. So what do these filoviruses look like? Cartoon form, we've got our RNA-dependent RNA polymerase, again, always present inside the virion because cells don't have RNA-dependent RNA polymerases, so they have to have a way of making messenger RNA as that genome comes inside the cell. The genome itself is bound by this nucleocapsid protein in a very helical form, again, incredibly reminiscent of what you see with the Paramyxo and Rhabdoviruses. Then you have a matrix protein. They have different names here, unfortunately, VP40, and membrane and membrane glycoprotein, just named as GP. The only real difference here is this VP30 protein, VP30 protein, which seems to be associated with the nucleocapsid protein as well, bound to this RNA. And exactly where it is in the genome is not entirely clear. So any of you who have downloaded the PowerPoint have had, oh, what the heck does he mean by C videos? Because I tried to insert them in here. If anyone's got a good idea how to get videos from Twitter and insert them into PowerPoint files, let me know, because I did want to take a quick look at those. Some of you may have seen this already. This is the video which I wanted to show from Twitter posting literally of yesterday, which I was not a huge fan of Twitter, and then I don't want these. But then I start seeing some of these things on Twitter, which I'm, wow, I have to stay on Twitter. So let's screen back here a little bit, replay this. So this is a 3D, I haven't tried to get the Oculus Rift goggles to take a look at it, reconstruction of what people think these monomegavirales, in particular the Ebola virus, should look like. So we've got a envelope, and this envelope glycoprotein sticking out. That's what these things are all supposed to be on the outside. And then it sort of goes through into the inside of it in this process. So we'll play this again. That's your envelope. Zoom into the envelope, and then strip some of this envelope off, get the matrix protein, They have the nucleocapsid protein as well as the RNA-dependent RNA polymerase bound there. And just like we had with all of those Paramyxos and Rhabdos, bound to every 6 nucleotides and protecting that RNA, still leaving the bases on the outside so they can be made. So that was the first one that I wanted to show here, and again, posted just yesterday. The other one was this one, which is a much higher resolution structure of the nucleocapsid protein bound to the RNA. And it's running a little bit quickly here, but the groove that hopefully you can see between these capsid proteins, or as I say, nucleocapsid proteins, is where that RNA is going to sit. So this is the RNA and that nucleocapsid protein of a short piece. It's not the whole thing of the Ebola virus internal structures. Okay, so let's go back and look at these. I will try and post links so people can take a look at these a little bit later. Yeah, Echo.
These viruses are coming pre-packaged with their own polymerase?
Yes.
Which is different than most of the other viruses.
Okay. Other than, which ones? What we talk about on Monday? Paramyxos and Rhabdos. So measles also has these. Yes. Any negative strand RNA virus has to come in with the polymerase already in the virion. So, and we'll see this is true also for the retroviruses right at the end of the term. But this is a big difference that you have to be bringing in enzymes with your genome. So of course, it also means that your entry process can't quite be the same. So you can't have something like a poliovirus or a bacteriophage that just makes a pore and the nucleic acid genome goes through it. You've got to have a way of getting much more inside the cell. Okay, but yeah, it's a very important point and bears repeating. Thank you. Okay, so what does that genome look like? Pardon me. Up here is our Ebola Zaire genome, and down here, just for comparison, is the Sendai genome. This is that classic Paramyxo virus, again, the one that most people have worked on. So again, Paramyxo viruses, 3' end to 5' end, again, negative strand. Got our structural proteins here and our major non-structural protein, the polymerase here. Same thing is true for these filoviruses. Nucleocapsid protein, the one that you need the most of. Other structural proteins, these guys are the matrix proteins, glycoproteins, similar to the fusion and your hemagglutinin neuraminidase, and then this regulatory protein, VP30, and then your RNA-dependent RNA polymerase. So the general genome structure is extremely similar here. Also have similar kinds of intergenic regions, like right here, where you have the end of one transcript and a whole stretch of U's. And what are those U's used for? Used for? Poly-A tail formation, because we have what? Stuttering that takes place. Thank you, John. So same thing is happening here between some of these, but you also have, and this is a difference between these filoviruses and the Paramyxos and Rhabdos, in a couple of places you actually have overlapping starts and stops for some of these genes. And so the start here of the transcript for VP30 actually overlaps the end of this GP, SGP that we'll get back to in just a second. Otherwise, again, stretch of U's, same deal, will still get stuttered and made into Poly-A tails. How do you not have stuttering take place? Because if you stuttered all the time, you'd have a problem with your genome, particularly if it's got to be a multiple of 6. Yeah, David?
High concentration of the nucleocapsid.
Exactly. Same exact kind of thing that's happening with your Paramyxos and Rhabdos. So high concentration of the NP protein that will cause stuttering to stop, and you just make the whole genome. Okay, so those are the sort of similarities, and then that one first difference between the two. We just look at the proteins. Not surprisingly, again, they're really similar. Nuclear protein, similar. Glycoprotein, this basically serves that same role as the hemagglutinin neuraminidase fusion protein that you see in the paramyxos. Those are fused in rhabdoviruses. So in like rabies virus, you have the G protein, again, glycoprotein, which is fusion and receptor binding all in one protein. And so these filoviruses also have that same thing, where you have the glycoprotein, which is serving very similar to the G protein, the rhabdoviruses. And then differences, VP30 that we'll get back to in just a second, directly associated with nucleocapsids, probably not binding directly to the RNA, because in that structure you could just see the NP protein associated with the RNA. And again, it also seems to be this rule of 6 process, where you've got 6 nucleotides and exactly 6 nucleotides bound to each of the nucleocapsids or NP proteins. So probably just associated on the outside. And that does seem to be important for messenger RNA synthesis. And again, we'll look at that in just a second here. And then VP24, I'm not quite sure why I made this box here. It's just an extra protein. But otherwise, all of these other ones are extremely similar in terms of their function. This GP protein, however, is a little bit different. And again, still seems to serve the same role as the G protein does in the rhabdoviruses, binding to receptors, membrane fusion taking place. As always, has a fusion peptide, which is the very hydrophobic piece of the protein that then will stick into the host membrane and cause membrane fusion to take place. Also generated by proteolysis. And so this starts out as being one big protein, so GP1 and GP2, and then there's proteolysis that takes place here. All of this is on the outside of the virion, and so also highly glycosylated. It's made through classic ways that you make membrane proteins through ER Golgi process. It's a trimer, just like so many of the others of these fusion proteins that we've talked about before. Why trimers, I think, is a really open and interesting question. I haven't seen a really good answer why trimers, but there do seem to be lots and lots of these trimeric proteins. So maybe there's something specific about that. And then one of the big questions was, what is the receptor for these proteins? And it took quite a long time to figure this out. But eventually, and it's actually kind of a, I don't know, fun story is maybe not quite the right term, but it was found that the receptor for Ebola viruses, at the very least, is this protein called Niemann-Pick C1. And Niemann-Pick C1 is a protein that is very important for lysosomal sorting and different fat concentrations inside different cell types. And what people had found is they were looking for things that the Ebola glycoprotein bound to. And they found this protein. It's like, oh, this is kind of strange. Why do we have this lysosomal protein? And what they wanted to show was it actually was something that was important in terms of getting this viral infection to take place. What was extremely useful as far as is concerned, why is it called Niemann-Pick C1? Well, it turns out that Niemann-Pick disease is a very rare genetic disease where fat accumulates in different cells and different parts of the body, particularly in the brain, and then causes all kinds of problems. But because there were these few patients who actually had these diseases, they were able to get cells from these patients and then mix them together with the Ebola virus or some of the Ebola virus proteins and see that those could then not be infected, whereas cells from people who did not have this Niemann-Pick syndrome could be just completely normally infected. So it was you didn't even have to make a particular mutation, and particularly making mutations in human cells is not terribly straightforward. And this was great, and they're not cancer cells, they're not HeLa cells, these are your normal cell types. So in fact, the rare diseases, yes, they're nasty diseases, but can also be incredibly useful in terms of understanding how the normal case ends up working. So this is just the crystal structure which they made of the part of this GP, glycoprotein again from Ebola virus. This is the piece that is gotten cleaved off. Again, this is the receptor binding part. The fusion part is the other part, binding pretty specifically now to this Niemann-Pick C1 protein. This seems to be mostly happening inside the cell because this is usually in the lumen of the ER, so somehow the virion needs to get inside the cell as well, and exactly how that happens is a little less clear. How do we put these things together? Again, check our lecture from last time. Start, stop. Again, go through your structural proteins, hit a stretch of U's, stutter because you don't have too much of the N protein, and then sometimes fall off, sometimes stay on and keep going. How you end up with genome, again, is just the amounts of your N protein, and then buds at the plasma membrane through assembly of these matrix proteins and the glycoproteins together with that. So the only other thing that I wanted to mention particularly here, and this is a difference between the filoviruses and the Paramyxo and Rhabdoviruses, is they have this editing which takes place, and we talked about that before when we talked about the Paramyxos and Rhabdos, the PCV sets of open reading frames where you have stuttering, which changes the open reading frame. Here, curiously enough, stuttering seems to be absolutely required to make this glycoprotein. And if you don't have stuttering, which happens here again in the middle of your messenger RNA, you end up with this secreted glycoprotein. The only reason it's secreted is because the transmembrane helices of the glycoprotein are all down here at the C-terminus of the glycoprotein. Now, having a secreted glycoprotein seems like a kind of crazy thing to do. If this is the protein you need to bind to your host cell and get fusion to take place, if it's floating around, then that's never going to work in terms of getting the fusion of your virion to the host. So in this case, unlike the case in the Paramyxos and Rhabdos, where that editing doesn't seem to be as crucial, here the editing is absolutely essential, because otherwise you wouldn't have the glycoprotein, you would never get fusion. So that's the process here. Now, why they make these secreted glycoproteins is a very open question. We don't know. But one hypothesis is that basically what's happening is it's sort of fooling the immune system. And the immune system is reacting with these glycoproteins, and then they're getting bound to antibodies, and then those are being used to pick up the whole rest of phagocytosis, et cetera. But all that happens then is just the glycoprotein, which is getting degraded, and not the rest of the virion. So that's the hand-waving theory about why these are making these secreted glycoproteins. Okay, oops, going too quickly here. And you saw that I'm going to have a clicker question. So the only other thing which is different between the filoviruses and the rhabdo and paramyxo viruses is this quote-unquote transcriptional regulation. So again, I don't like the transcriptional term because this is all RNA-dependent RNA polymerases and doesn't have to do with DNA-dependent RNA polymerases. But what was seen actually relatively soon after they sequenced some of these viral genomes is that there were these hairpin structures which seemed to form at the beginning of actually almost all of the messenger RNAs, but also the genome of all of these filoviruses. So as soon as, again, the genome is always bound to the NP protein when it comes inside the cell. And so the RNA-dependent RNA polymerase will go through that and make a messenger RNA. But that messenger RNA forms a secondary structure, and a strong secondary structure, right at the 5' end. And so that both seems to block further messenger RNA formation, but also, not surprisingly, is going to block translation. So we talked about secondary structures before, way back when we talked about those RNA phage. So secondary structures seem here to be very important in terms of regulating not just translation, but also further messenger RNA production. And VP30, at least in Ebola virus, seems to be very important for allowing these to be made. And probably what that is just a way to make sure that the cell is going to be making the viral proteins more so than it's making its own cellular proteins. But exactly how that regulation works is not completely straightforward. And part of that has to do with the fact that these are really nasty viruses, and so you don't necessarily want to work with them. And if you do, you have to be under very high biocontainment facilities, which we'll take a look at in just a second here. Okay, so now finally, what are we going to do? Ask our clicker question. Okay, so what did I do with my... Turn this guy on. So which of the following is a major difference between measles virus and Ebola virus? Polymerase stuttering, order genes in the genome, poly A tail formation, regulation of mRNA production, or the presence of glycoprotein? Ten, 5, 3, 2, 1. So either somebody changed their vote at the last minute or they want to talk again about it. So discuss with your neighbors a little bit what you think the answer is, and then we'll vote again. 10. I should, hopefully everyone's seeing their clicker scores. I should have posted all of them. If you're having trouble seeing them on D2L, let me know. Okay, what do we think? I agree. Yes, it's that regulation. Everything else is extremely similar. If I'd put here the presence of secreted glycoprotein, that would have been a different story. But just the presence of glycoprotein, that is... That's fine. So select our answers, close. So wanted to sort of, again, switch gears a little bit. We're kind of done with the molecular aspects of things, but did want to mention a couple of brief things. I did mention that TEM image of the Ebola virus virion. That was taken in this building right here in Galveston, Texas. Kind of looks like a bit of an office building. All of these windows here actually go into a wall. There's actually not real windows here. And you can kind of see this block right here in the middle where they don't even have any windows at all. This is actually a BSL-3 lab, biosafety level 3 lab, with the biosafety level 4 lab in the middle of it. And that's pretty much always the way that these biosafety level 4 facilities are set up. The biosafety level 4 lab is actually inside a biosafety level 3 lab. And we'll take a look at a video on this in just a second here. But it's in this, in fact, BSL-3 space where there's an electron microscope, and I think it's the only electron microscope in the world where they have an electron microscope in the BSL-3 space. Partly to mention this is the only place you can do these kinds of experiments, which is, how do you put together one of these infectious negative strand RNA viruses, because, as Echo pointed out, you have to have the polymerase associated with the RNA. You have to have all the proteins associated with the RNA. How do you get that to happen? What you need to do is have lots of different plasmids which will express all of the proteins. Then you have to have a genome sequence which can be made with our friend the T7 DNA-dependent RNA polymerase, and so that will make your genome. You have all the proteins together, and only when you have all of the different proteins and the RNA together in a cell, this can then get assembled and eventually then get out. So these are really complicated experiments to do, and to do them under these high containment experiments is really not straightforward at all. So wanted to show you a little bit about what the inside of a BSL-4 lab actually looks like. This is there's a really nice, actually it's like an hour-long TWIV, which is a video going into a BSL-4 facility which just opened in Boston. They were very fortunate to be able to go in before it actually went live, or hot as they say. They didn't actually have any of the pathogenic organisms in them. But then there was a Canadian Broadcasting Corporation documentary that came out a little over 10 years ago now, where they also go into the BSL-4 facility in Canada. And so we can take a quick look at that. That would be here. We can escape. Stop this briefly. That would be here. Hopefully we'll be able to hear the audio on this as well. This is a production which they did. Let's see, full screen here. No. Full screen. One of these will look up. So this is, again, that's a documentary that the CBC did on the virus hunters. And if we're lucky, we'll get to see a certain virology professor from Portland State who's also featured in here later on. So this is, just the beginning here. Do we have any audio? No, the audio is not happening here. It's supposed to happen with HDMI? Why is it not doing it? Oh, well. So this is the entry to the BSL-4 facility in Canada. As far as I know, there's only one, which is in Winnipeg. And then this is the interior camera. First you go through an airlock, and you have to put on your bunny suit and double, triple glove. Everyone is attached then to an external air source. And then once you're inside, you have to do all of your experiments when you're all gloved and kitted up in this way, which is really a very difficult way to do any kinds of experiments. And here is the virologist, in fact, looking at some of his cells. I don't think these are viruses. They're trying to pretend that they're viruses, but they're really not in this particular case. So not quite sure why the audio didn't work on that, but I'll work on trying to make sure that it happens again later. They also have, and partly why this was put in here, the Canadian Broadcasting Corporation also had some pretty amazing footage of a Marburg virus outbreak in Angola, and the virologist who was shown there going into his lab in Winnipeg was also then on site in Angola going through the whole process there. So Marburg, BSL-4, Marburg virus. Why Marburg virus? And certainly this area doesn't look terribly much like what I just showed you the pictures of in Angola. So Marburg virus is a virus that was identified in Marburg because they were working with monkeys. And so a couple of people got sick, and they got sick rather precipitously and had hemorrhagic fevers. In the 1960s, and there were about 60 cases of this particular virus. And then there were also other cases of monkey handlers elsewhere. Probably this was a disease that had been known much longer than that, but hadn't been recognized by the Western Europeans or North Americans until they had this particular outbreak, and it just ended up being named Marburg because of this. Most of the people who you visit in Marburg, have actually been there and given a couple of seminars, are not wild about the fact that there's this hemorrhagic fever virus named after their really beautiful community. This is the castle in Marburg, right, sort of in the top of the town, right next to the medieval old part of town. So, but named because, again, this was where it was quote unquote discovered or identified. Most, however, of these hemorrhagic fever outbreaks until very recently, a few years ago now, were really in Central Africa. And so, in fact, this is the Marburg outbreak that we have the footage from the CBC, was in northern Angola. Ebola, and actually very well described here in this No Time to Lose book, was originally isolated in this area here near the Ebola River. And in fact, they didn't want to call it the Yambuku virus because Yambuku did not want to be associated with this particular virus. So the Ebola, there's actually the Ebola River very near here. So this was the first example then of Ebola virus. Marburg had actually been found, quote unquote, earlier. But then at that point, they knew kind of what they were looking for, knew they were looking for these filoviruses, knew some of the sequences, knew some of the symptoms. And then various outbreaks happened, again, almost all of them in Central Africa, with a couple of exceptions outside of Central Africa. About 500 cases until 2005, and very much self-limiting. So there will be a disease outbreak. It would end up being relatively self-contained, and we'll see why that is in a little bit here. And then would die out, flare up, die out, flare up, etc. And then there was an outbreak in Reston, Virginia. Reston, Virginia? What the heck? So that actually was in, yet again, so Reston, Virginia is quite close to Bethesda, Maryland. What's in Bethesda, Maryland? Military bases is one thing. What else? I always go and beg for money. NIH. So, and a lot of the, actually more so the NIH than the military facilities do lots of work on non-human primates. So the non-human primate center for many of the NIH and military studies was in Reston, Virginia, and a bunch of their monkeys started to bleed out of all of their orifices, and this was clearly a problem. So they then identified that it was, in fact, an Ebola virus. Fortunately, did not infect humans, and this turns out to be very, very useful if you're going to then start to be interested in how Ebola virus functions. If you can work on something as a human that doesn't infect humans. Don't get it around the non-human primates because they've got lots of problems. So very nice model system to study Ebola viruses is this Reston Ebola virus. So why does everybody care about Ebola viruses? Yes, it's ugly because of the hemorrhagic fever, lots of bleeding, but the major thing is a very high fatality rate. And even now, even in the current cases, although we're talk about the vaccine, which is still experimental and not allowed officially to be used, anywhere between 30 and 90% fatality. In fact, in the latest outbreak, I think it's about 60% fatality rate. So if you get Ebola, it's really very, very bad. It does seem to mostly infect macrophages, which is a classic issue with the diseases that cause all kinds of problems, because if you, again, the virus, infects the cells that are supposed to be protecting you and blasts open those cells, of course, you've got a real problem in terms of fighting off not just the viral disease, but also any other diseases which happen to be there, and particularly in places like Central Africa. There are lots of other diseases which are present there as well. So not just the Ebola virus. It does seem to be a zoonotic disease. We haven't really talked too much about zoonotic diseases, but we got to talk about reservoirs when we talked about the coronaviruses and SARS. The idea here is that there's a virus which is circulating in animals which are not humans, and then at some point there's a crossover into humans, and then disease will happen in those. It's probably not the right host for these viruses. And again, think about it. If you're dependent on a host and you're killing off all of your hosts, it's not a very good way to be able to persist. So lots of these nasty diseases are actually really not very good from the point of view of the virus. How do you get infected? Turns out that it's very close contact with these viruses, and we'll see why that is in just a second here. Very close contact with someone who has had the disease, and one of the things about Ebola in particular, in Marburg too, is there's very high viremia, and viremia is something that Dr. Hirsch talked about before. Very high amounts of virions that are produced in patients, someone who's actually is diseased. And so these very high viremias means that the direct contact turns out to be a way that you can end up getting the disease. And unfortunately, what this has meant is, particularly in early parts of outbreaks, medical personnel are the ones who have the very close contact with the patients, and often they're the ones who then come down with disease. And particularly early on in the West African outbreak that we'll talk about in just a second, a lot of the medical personnel were the ones who were dying, which is a real problem if you have this disease which is spreading like mad, and then all of your medical service providers are getting sick as well. It's really kind of a vicious circle. The other thing is that the media get way, I don't know, out of control, maybe it's a little bit too much. But this whole hemorrhagic fever being nasty and high fatality rates, but still very few cases until 2013, under 5,000 cases. And so 2014, again, 2,800 reported infections, 1,900 deaths. So high mortality rate, but actually relatively low numbers of cases. And still actually very low numbers of cases. As you may have remembered measles, when we talk about measles, there are millions of people who are getting measles-related diseases as opposed to thousands here. Yeah, go.
From an epidemiological standpoint, because it has such a high mortality rate
Mm-hmm.
and it kills so quickly, like once you start transcription, pretty much it probably wouldn't spread like flu.
Yeah, well, so
It's not, you're not going to see really high amounts of infection like spreading.
yeah, so the rapidity of spread, and measles is probably even a better example. So yeah, no, it is actually pretty hard, apparently, to get infected by Ebola. It has to be very close contact with someone who is highly viremic.
And that person's probably not going to be walking around in public for too long.
Probably they aren't going to be able to walk. And some of the stories in terms of the West African outbreak is they're almost always being brought to the clinic because they couldn't get there themselves. Again, getting back to the epidemiology, however, there only seem to have been actually less than 100 cases of the virus getting into humans in the first place. So coming out of that animal reservoir, which is, actually I should have mentioned that here, our friends the bats here, which are the ones which are the natural reservoir for these viruses. And exactly how that transfer takes place is, again, hard to tell. But if you look at, and one of the other things which has happened, particularly with some of these later outbreaks, is you can follow the sequences of lots of these viruses, and you can see that the genome sequences are different in each of these different outbreaks. And so you can pretty easily trace, there was one introduction that happened here, those are the viruses which spread in that particular case, another introduction somewhere else. So relatively low numbers, and the virion's a wimp. It's heat, detergent, light, labile, so leave it out, and you really aren't going to get much in the way of infection. And this is actually then very different from measles, because measles stays infective for long periods of times in aerosols, etc. So actually very fortunate, even though molecularly these are really similar, probably it actually has to do with the length of these virions. They just don't hang out in aerosols very well. And so, if anything, very lucky to do that. And it's actually pretty hard to find viable viruses, or at least it was. Almost always it's going to be bodily fluids where you can find these virions. And you have to be pretty careful about them, and otherwise they will fall apart. So this is maybe getting back to your hot zone. The media got very excited about this. This was, even back when there were 3,000 cases. Hot zone, Dustin Hoffman, you may be able to recognize here, out in the environment and trying to save everyone's lives. Not a very good film. I can, if we've got some more time, maybe over some beers after the end of the term, I can tell you all the problems with this particular film. But that was basically where we stood when I gave this lecture. It was the 19th, so 7 years ago. You had a few cases, and this was in 2012. May 2011, 1 case in Uganda, 7 cases in Uganda in December, 24 cases in October, and in the Congo, 77 cases. And again, 50+% fatality rates, but all seem to be really pretty limited. And interestingly enough, so this was the 2nd of May, 2014, so 5 years ago, I was preparing this lecture, and there were these cases in Liberia and Guinea, 234 cases, 146 deaths, 62% fatality, lots of them healthcare workers. So what was going on? This was strange because almost all of the filovirus outbreaks to date had been in Central Africa. And so what were they doing in West Africa? This was very, very strange. And again, this is the 2nd of May, 2014. So 14th of April in 2014, going back a little bit, this is where you were having these outbreaks. So for those of us not familiar with African geography, which I wasn't terribly familiar with, again, all of the previous outbreaks had been here in Central Africa, and now we're way over here in West Africa, particularly Guinea, Sierra Leone, and Liberia. And this was, again, in April, where they had a few cases, and unfortunately, many of those cases here in very underdeveloped areas of Guinea, Sierra Leone, and Liberia. Unfortunately, also an area where there had been lots of civil war and really not very good infrastructure in that particular case. And particularly this area right here, Kisedugu, this is the typical marketplace that you have in Kisedugu. Lots of people, main transport here are motorbikes, and this is actually the main sort of urban center that you have here, where people think that the original crossover event that took place in this West African outbreak is Meliandu. This is the main road into the village here. Most of the transport, so it's a little bit hard to see here, but is just by foot and occasional motorcycle traffic. You can see the motorcycle here being repaired. And it's right at the border between Guinea, Liberia, and Sierra Leone. And most of the people there didn't really care too much about borders. You go across one way to collect your food, you come back, you sell your food in one place, you come back, and very far away from any kind of medical support. So what happened there is there was clearly an outbreak, but nobody knew about it for, in fact, quite a long period of time. And unfortunately, it started to grow and get bigger. And what happened here is there were a few cases in Guinea up through the beginning of 2014, and then all of a sudden there was this big burst. And a lot of this burst actually happens because people were now going to some of these urban centers and going into the hospitals, where they then were contaminating, getting some of those medical providers sick. Why was that? Because there wasn't any filovirus disease in West Africa. So none of the local healthcare practitioners knew about this disease. So this was a big, huge bump. And then, as it happened in almost all of the other cases, and I pointed out before, the case numbers went down. So, right, we're fine. We're not in too much of a problem. But then, unfortunately, people then started moving to some of the bigger population centers, and there the disease spread further and further. And so there was this second bump. And hopefully what you see here is the number of cases here is still relatively small. 30 cases confirmed, suspected, probable, up through about June of 2014, which turned out to be a real problem. And a lot of it was political, a lot of it was not believing again that you would have Ebola virus in West Africa. Everywhere else the disease was self-limiting and disappeared. Here, that turned out not to be the case at all. And so now we're talking thousands of cases, again, starting right here between Guinea, Sierra Leone, and Liberia, literally right in this corner. That's where Kisidugu is. And then the big problem was that this started to spread to the cities. Konkari, which is the major city in Guinea up here, in Sierra Leone and Liberia, got into the cities and turned out to be a real huge problem, and thousands now of cases of Ebola, and people just had no idea what to do with them. And eventually what happened, and most of this actually has to do with PPE, and so personal protective equipment, and making sure that there is not direct contact with any of the people who have Ebola, which is really tough, and it's unfortunate we don't have time to look at that CDC video, but there's a lot of lack of trust of the medical people, the non-local people, and as soon as they start dressing themselves up in these gowns or really a PPE equipment and completely separate from the patients, this is kind of anathema as far as most medical treatment is concerned, particularly medical treatment in the developing world. So there was a lot of resistance also to getting treated, going to some of these treatment facilities. The person who brought in the sick Ebola patient would come in on their motorcycle, drop them off, and leave. The problem is that person has clearly just had very direct contact with someone who is probably highly viremic. So where have they gone? And you need to trace them, make sure that they are okay. And that whole process takes a lot of time, takes a lot of people, and people are scared. People are scared and they hide. And they don't want to end up in these places where many people die. You go there and you die. Well, that's not a really good thing, is it? But there are some people then who recovered under those conditions, but that was much less obvious. So to make actually a relatively short story, also short too, through 2016, you remember there were about 3,000 cases of Ebola before this. Now we had 15,000 laboratory confirmed cases, probably more like 30,000, and 11,000 deaths. So massive outbreak in West Africa that really nobody was prepared for at all. And so hopefully we're getting a little bit better about that. So I need a break. Which means I'll ask you a question. Hopefully everyone will get this because I've been beating on it so much. Before 2014, most Ebola outbreaks were in which geographical area? West Africa, East Africa, Central Africa, South Africa, the USA. I didn't say Ebola hysteria. That would be a different answer. Still aiming for some more 100% here. I probably should have put up a map what I meant by all of these different regions. But 5. Okay, we're not quite at 100%, but we're getting close. So yeah, almost all in Central Africa, and that was potentially why the outbreak in West Africa took so much longer for people to be ready to deal with that process. So why did I put the USA on here? Because there were 4 cases in the US, 2 of which were people who were infected in West Africa and came to the US, and 1 person died, and this is in fact one of the patients here being transported from the hospital where they were in Texas being transported, and again they were healthcare workers who were the ones who were infected and then were moved. And 1 person, in fact, died, and that was, in fact, that very first person who came into the U.S. in Texas. Even though that was the case, this was some reporting, that was actually before this particular person had died, Americans concerned that they or their family will contract Ebola. 51 million people were concerned about Ebola. And my primary care physician said that people would come into her office and want to get an Ebola vaccine and then said, oh no, I don't need a flu vaccine. Okay, so we'll talk about flu, don't worry, on Friday. So yeah, this got really kind of out of control. That being said, there were actually some spillover from the West African outbreak. And again, there were 4 cases in the US, one of whom actually died. But what I'm particularly impressed by here are some of these other ones, Nigeria, Senegal, Mali, where they did have a number of imported cases, people who had come there from the endemic areas, Guinea, Sierra Leone, Liberia, who were then immediately diagnosed appropriately in the locations where they were and isolated, treated, and there were relatively few deaths in Nigeria, none in Senegal, few in Mali, but still very well self-contained. And so the responses of some of those local healthcare agencies were extremely good, and particularly in Nigeria, which is a very populous country and very urbanized. They did an extremely good job of controlling the outbreaks which are there. Although some of you may have noticed that Nigeria is considered more as Central Africa, and people actually did know more about some of these filovirus diseases. So this was, again, because of this outbreak. One of the things that in fact happened through that outbreak was people kind of went back into their freezers. And actually, some of those close to where I showed you that video from the BSL-4 facility in Canada was a Canadian group that had actually come up with an Ebola virus vaccine. But nobody had cared about it until this outbreak happened. And interestingly enough, it's a combination of the vesicular stomatitis virus, which is one of those rhabdoviruses that we talked about last time, where they just put the glycoprotein of Ebola virus on it. Vesicular stomatitis virus, no disease in humans, but you put this glycoprotein, you get a really nice immune response. So this was then tested in West Africa and elsewhere. I just wanted to briefly go through this sort of how you do clinical trials. Phase 1 trials are all about safety, where you had 20 to 100 people. And these are actually not in areas where they had disease, because you don't want to have the disease there. You're just talking about safety at this point. Phase 2, hundreds of thousands of people. This was done in March 2015. Unfortunately, March 2015, remember the outbreak was basically almost done by then. And so they were looking at some people who were then actually in these regions. And so this was then in West Africa. And then finally at phase 3 with thousands of tens of thousands of people, this is actually start thinking about efficacy. And so they did in March 2016, so actually very little amount of Ebola virus disease was still present there at the time, but they saw these really high amounts of efficiency, 70 to 100% efficiency of this protecting against viral disease. And so one of the things that I saw the other day, apparently the, I call them pro-disease people as opposed to anti-vax, want to see as double-blind clinical trials. You want to sign your kids up for one of these Ebola ones, right? You don't know if they're actually protected or not, and then you're going to expose them to disease with a 60% death rate. Try that argument with people who are pro-disease at some point. So, but there's actually now a vaccine that works very well, but it still hasn't been approved yet because we haven't gotten all of the studies here go through to approval. However, it's been now used in the latest outbreak. How many of you know about the outbreak of Ebola that's going on right now in the Congo? Second largest outbreak ever of Ebola going on in the DRC right now. As of Monday, 1,500 cases, 1,000 deaths, all going on here in eastern and particularly northeastern, sorry, it's a little hard to see here, this is Uganda and the Democratic Republic of Congo right here. We have now a vaccine that works really well against Ebola. How is this still spreading? This is the reason that it's still spreading. There are 13, at least, different armed groups which are fighting over mostly components of cell phones, which are present in this part of the DRC. And because of that, it's really hard to get the vaccines to the right place. And unfortunately, about a month ago, there was a Cameroonian epidemiologist who was actually talking about vaccines in this particular area who was shot dead while he was doing his presentation about why this was a good idea. And again, many people are scared. There's not very much confidence in the government in this part of Africa, and the numbers are still going up. So we need to be, I think, much more concerned and interested about what's going on here. So yeah, everyone got very interested in what's going on in, excuse me, in West Africa, but now in Central Africa, much less so. And this is, again, even in the case of where we have quite a lot of this vaccine, which seems to be working actually extremely well in these cases. It's just getting those vaccines out to the people who really need them. So it's not really a medical problem here. It's a sociological problem. It's a political problem to actually try and deal with some of these diseases, which are much harder problems to crack. So on that wonderful, uplifting note, just wanted to mention a little bit more about where people think that these viruses are coming from. Again, probably circulating in the bats in a regular reservoir cycle. People call it the enzootic cycle. Again, doesn't seem to be much in the way of disease. Occasionally, there's some kind of spillover, probably in many of the cases actually through non-human primates or even some of these antelope, and then those then transmit to the humans, and they then transmit between themselves. So bushmeat is also potentially a major reason why you have some of these diseases. And in fact, one of the cases of Ebola virus, well before this, was a chimpanzee hunter who came down with disease soon thereafter. So if you care about what these symptoms are, these are some of them right here, but we're at 10:05 already. And so I think we'll stop here and then talk about actually a much, much scarier disease on Friday.